I believe what I find most fascinating with Sean is his way of finding rigorous solutions that people can, in fact, understand. So in some sense, you started this idea of visualization before you talked about visualization. We very early on had discussions where Sean really tried to always get down to the point, how can I make it come into the head? Not how can I find the truth only, what is right, but right has also always somehow to do with show. And I believe this is what he sort of has taken in WSBI, visualization in biology, onto a new level. Sean is completely changing the way computational biology is approaching the issue by pushing this idea of visualization is important for science. And Sean is in fact the first who, in some sense, made, or one of the first clearly, worldwide, who made a life out of it. So you in some sense have become, you still root in the history of Proteins, and I pushed him because of personal interest into giving a talk today about the dark proteome, which is in some sense mostly back to the science he did before, but in some sense I believe your major breakthrough achievement, what is your niche, what you stand for, is really that you teach us all how important visualization is, much beyond the level of, yes, we understand that we can't see it. And this is an amazing achievement. Thanks.
Okay. Well, I hope I'm allowed to say a few words about visualization as a prelude. So I was planning to talk a little bit about Viz, just to make you aware of some resources, and then talk about Aquaria, some of the new things we're doing, and then lead into the dark proteome, because dark proteome is kind of what Aquaria doesn't find. So I'll just really quickly jump through these slides, because everyone kind of knows the basic idea that we have massive amounts of data, right? This is cost of sequencing falling hyper exponentially compared with the cost of computation falling according to Moore's law. Those of us who've been in bioinformatics analysis know that to completely analyze the genome is actually slowing down, because our data and analysis, the amount of data we have to process is increasing faster than computer speed. And so we have just nucleotide information alone, over a terabyte a day being deposited in the short read archive. We have over 2,000 original scientific publications in the life sciences alone. So data tsunami. What are we going to do about it? Very common human reaction is, okay, I'm just going to focus on my thing and I'm not going to worry about that. But that's a pity when you think about it. There's undoubtedly already discoveries about fundamental diseases out there in public domain data unseen, because we're all just too busy with our own thing, and those data aren't organized, and we need to get more serious as a community about how we organize and how we view those data. And I bring good news that we're all born with an amazing pattern recognition system. Over half the human brain is dedicated to visual processing, or heavily involved in that. And here's the thing. Here's the vision. If we can learn to encode our complex and abstract data, I don't think it's going to work, but all right.
Yes, it is going to work. Don't worry. Just take continue. Don't worry about it. Don't take it seriously, and it will work. And just get out of it. Yeah. And now you're there.
All right. Okay. So, yeah, if we can learn to encode the data that we're dealing with, which is usually abstract data, doesn't normally map onto an obvious spatial arrangement or topology, if we can learn to encode our data in ways that work together and well with human vision, we can start to take advantage of this processing unit that we're born with, and start to see more and get more from our data. But here was one of the big insights for me. There's a couple of ridiculously stupid cartoons, but they are actually concept shifts that happened in my mind. About 2000, 7 years after we met, I was thinking, okay, we got to visualize stuff. But then I had another concept leap, which is, it's not about putting the right stuff on the screen. You can have the greatest visualization you want, but the system that we're designing for, with predict protein or any bioinformatics methods, involves a human in the loop. A human being has to see and understand the data. They have to know how to manipulate and work their way through the data. Otherwise, it's a tree falling in the middle of the forest. You can have the greatest visualization, but if no one sees it, then there's no insight, there's no discovery. And the point of data visualization is not about making beautiful pictures. A lot of people, and you'll probably see nice images, nice graphics here, that's exactly not what we designed for. What we designed to do is to make the data itself clear, and when you look at the data itself, the beauty or that you see, the aesthetics that you see, is the data itself, like a protein structure. Get rid of everything else as much as possible. So the purpose of visualization, particularly if you're using it in bioinformatics context, is to enable this light bulb moment to create insight. And so, as Burkhard mentioned, one of the things that we did back in 2010 was we started this meeting, and the idea is that we realized that we've got data visualization happening across all these different areas of biology, across genomes, transcriptomes, proteomes, cell biology, tissue biology, and populations of various kinds. And we've all got our own tools, and you guys are sort of more in the protein space. When does someone who creates a system for visualizing genome data, when do they get together with someone who creates a tool for visualizing MRI imaging data on tissues? They never normally meet, and ideally these tools would integrate, right? We'd have sort of this vision of systems biology, all of our tools integrating, interoperating. How are the tools going to ever talk to each other until the people that make the tools talk to each other? And there was no meeting where the people who make the tools that biologists use to visualize their data actually meet. And so we created a meeting. And in addition to biologists and bioinformaticians like yourselves, we invite specifically a bunch of keynote speakers. And these are from outside of biology. And you've all seen some enormously fantastic data visualization from TED Talks and so forth, right? And out in computer science, there's people doing some really cutting edge stuff. They're not in biology. And what we realized is if we wait for those guys to invent some beautiful biological visualization system and come and bring it to us and say, here it is, here's the solution, it's not going to happen. We've got to go and get those guys, bring them, drag them into our community, get them to meet us, form collaborations with them, and that was the idea of VisBee. So this was the 7th meeting we had just last week, and we alternate every year between US. Actually, it might be good to get the lights down or to drop the light a bit, because there's going to be a few slides like this where, I don't know if you can see, that's a NASA image of the night sky.
It's not working. It's not working in the other one as well. So.
Weird.
All right. So what about these guys here?
This I'm going to do it. But
yeah. Well, a little bit. Actually, probably a little bit is going to be enough. So it'll improve it. Slightly better. Okay, so we just had the 7th meeting in Heidelberg, and I always say, it's not as if I'm trying to tell you about this great meeting that's happening that you've never been to. We've put a lot of work into making the outcome of the meeting accessible, so we've really spent a lot of personal hours making this website. You can get all the posters in Zoomable form. We also have videos of almost all talks, and particularly the keynote talks I really recommend looking at. We've tried to get really the best, and some of them are really exciting, really useful. I particularly recommend the keynote from Tamara Munzner from 2011. She literally wrote the book on data visualization, and she's just not only an expert in that field, but she's an expert in explaining it, and it's a really great talk and a really great book. We did this special issue of Nature Methods in conjunction with the first meeting, and this had a huge impact on the community. So this basically was just a compilation of a review of the data visualization methods and tools being used across many areas of biology, and it clearly showed that we were addressing a need that essentially no one else was meeting. I'm quite proud of this outcome from VisBee, and particularly happy because I didn't have to do any work after having made the meeting. The Nature Methods editor got together with some of the other keynote speakers, and every month in Nature Methods, pretty much since the first meeting, there's been a one-pager describing one aspect of visualization, like how to use colors or how to use layout. And now there's a collection of those things you can pull up, or just go manually pull them up from the Nature Methods beforehand. I mean, you can read one page, right? And if you're going to design a figure, or you're going to design an interface or any kind of visualization, definitely worth checking out those. Easy to access, you can probably get them for free. So now I'm going to talk about how we've been using these principles of data visualization in Aquarium, and just briefly run through it. So I was, as Burkhard mentioned, an ex-physicist, and I fell in love with the idea that in order to truly understand biological systems, we've got to understand the physical nature of the components, of the mechanisms. And of course, there's no greater example than this, but this is information storage and replication. We have thousands of other processes in the PDB. We have now over 16,000 structural entries there now. But that gives us a new problem. Just how the hell do you navigate your way? How do you find the right structure? And what I was always dreaming of, and this is going back now to 2000 really, was a Google-like interface that would just, for each protein, summarize all of the structural information that was either for that protein or anything similar to that protein. Put it all in a nice, simple environment on one page, and that is essentially the goal of Aquaria. And I think this is one of the themes I'm going to talk about a few times, is that data visualization is often about what's not there. It's about what's missing. You try to visualize something, and you see, and when you do that, you notice the gaps. And here, this was a sort of a big thing. There was no page that brought all those information together. And so with first SRS3D and now with Aquaria, we worked at creating that page. So I briefly mentioned, because of Burkhart's work, the sequence structure gaps. So we know that sequencing is going like gangbusters. And for fun, I sort of plotted this back of the envelope calculation, the fall of the cost of determining a protein structure on more or less the same scale. This is very back of the envelope-y, just looking at number of entries and comparing how it falls. Clearly, this might change in the future, but it doesn't seem to be a very near-term perspective. It's just enormously much more difficult and more expensive to isolate, purify, and resolve the structure of a macromolecule than it is to sequence it. And so we have a massive gap. But of course, structure is more conserved than sequence. And I actually we've been promoting around the world Burkhart's famous paper. Hopefully, many of you know that if you take all the structures in the PDB, look at all the structures that are identical and superimposable, the majority of them have a sequence identity of around about 5%, which is essentially random. So, and most people think of the other way around. They think that's kind of an exceptional situation. It's not the exception, it's the rule. So that gives kind of hope for us who are trying to predict structures, which is that many of the structures we need to predict are actually probably already in the PDB. And this was Andreas' work now, was when we started the new version. At one point, we looked around for a method that could automatically match sequences onto structures. And it seemed that the best method in 2010 was this HH-Blitz method. And so we started calculating our PSH2 database. And then a couple of years later, when the next CAST results came out, this method had slipped to 7th place, which initially sounds bad, but when we looked at those data on top, we realized that all of the other methods that would automatically map sequence to structure were all using HH-Blitz as a seed, and they were all at least 370 times slower. And since what we had in mind was a massive screening, then it was very clear that HH-Blitz was the best trade-off of speed and accuracy. I'm going to skip the next two slides because everyone knows this. And so I think they've seen your receiver operator curve. I think this is amazing, and I just pointed out that I just recently saw a receiver operator curve for the prediction of the location of transcription factor binding sites. And we're getting 83% recall, we believe, at 1% false positive. And I mean, in principle, predicting a 3D structure would sound like a more difficult problem to me than trying to predict where a transcription factor binds in the genome. The fact is, we've been doing structure prediction of proteins for decades, and with CASP and with all the attention that's been getting, we're getting pretty damn good at it. And we're orders of magnitude better at this. The receiver operator curves for transcription factor binding look just lousy. So the idea was run this across using all this PDB structures and in generating 50 million sequence structure alignments. And I think it would be irresponsible if we were just to do this and dump the 50 million models onto the community and say, here's all sequence structures aligned. To my mind, that's contributing to the confusion of the world, not the clarity. And that's the opposite of the reason that I became a scientist. I became a scientist to try to make things clearer, not make things worse. And I'm not saying we should stop as a community gathering data or modeling, but we have to get more serious about what we do with those data. And then there's the thing about the PDB. So I'm one of the greatest fans of the PDB. I think it's literally probably the best scientific database that I know of in the world. We've had stable identifiers since 1972, and it's amazing how early we adopted the convention that if you want to publish a structure, you have to have your entry deposited in the PDB. There's no other scientific discipline that I know that has a database like this. But it's created for and by structural biologists. It has its limitations. And if you look at a PDB entry, what got center stage on that page. The title of the entry, the list of the authors, their affiliations. The data's buried over to the side here, and then you've got like space group symmetries. A biologist who wants to use a structural data to gain insight into function is not interested in almost any of this, just maybe that little thumbnail. So what we tried to do in the query is put the data center stage and really focus on designing the whole system so that it's easy for people to find structures, and it's easy for people to use structures to gain insight into function. Now, I'm having problems with the internet, so I don't think I can run a demo.
Take Rust 2. Take Rust 2 here.
And it's the same,
Is the other one,
which I'll have to type in.
Yes, you have to type in right. So you have to type it in also because it's not an option. If you say join other network.
Join other network. Isn't it great when it's all lowercase?
Two, yeah. And then you say subsecurity, which one?
yeah. Right.
Rust lamp.
I'm not going to ask about the lamb.
I'm not entirely sure the one who did it. Is anyone able to answer the question? Okay. Please answer it to me before you leave, so at least one remains.
Okay.
I don't know the answer.
I would like to know too.
While the query is loading.
So they never told me.
Right.
Should I tell? Wait here?
We are online.
Okay, so this is hopefully going to load.
You see, Tim, this is what I meant. Somehow you're left of the screen. You're left.
No, it was justifying sometimes.
Huh? No. What? So the projector is not centered with the screen. It's not aligned with the screen.
Yeah. Okay, so I think I can continue. So we aim for Google-like simplicity. Just type in the name of a protein, and then one of the key things was each protein. There's many matches. I've seen the worst case I've seen is about 10,000 structure matches. This is for a sequence where there's just many entries in the database in PDB. In this case, 130 odd matches, and so we're using graphic design, data visualization tricks. For example, in this case, there's 83 structures that match in this part of the sequence, and we cluster them, and then we make the different structures available in what's called a tree view. And what we do is, there's 87 PDB structures. There's still a lot to look at. You know, at least they all match to that particular part of the sequence. And you see here that, for example, some that have 4 copies, a tetramer of P53, 4 copies of the molecule bound to a DNA dimer. That's actually kind of the more physiologically relevant states. You can sort of zoom down to individual PDB entries. And there can be a very large number of these, so we sort of have this sort of paging thing that sort of scrolls through them. So just, as I say, tricks to make this summary of all the structural information available, condense it down so that you can gain that overview, but still navigate to any particular structure. And the query, it's a work in progress. This is not the absolute final thing. There's more we want to do with it. And the second stage, of course, is mapping on features onto structures, and really the reason I created a query was this, to be able to click on a feature set. These could be predict protein features or something, and just map them onto the 3D structure. And from my mind, those are the two questions that most biologists, not structural biologists, but normal biologists are going to have. First, what structures are available for my sequence? And then, you know, which oligomeric state, are they bound to DNA, bound to some other protein? And having found a structure that I think is the most relevant to my question, how can I now learn about that? And I think just mapping of features is the #1 way that most biologists are going to learn from them. There's lots of other things one can do, and there's programs like PyMol, Chimera, VMD, and so forth that are specialized in doing all kinds of things like electrostatic field, and Aquaria specific does not do that, because we're designing for people who just want to occasionally look at structures. So that was Aquaria. The demo works, so I can skip the next few slides, which I'll just do. And so there's our API that enables you to just map any given set of features onto Aquaria. And Andrea has a really beautiful example of that now, mapping the SNAP2 features on, which I think, have you presented that already to the team? Or?
I presented the SNAP thing, but
right.
I haven't seen the project.
Okay, right. Okay, so you've seen that. So just to mention a few things that we're doing going beyond Aquarius. So Aquarius is designed to answer really simple questions. How do I find structures and what do I do when I get there? And the basic answer to that is map features on top. But let's suppose I want to do more complicated things, like look at all, in this case, this is an ensemble of all P53 structures. That's a really interesting ensemble because it's very heterogeneous. Some of those structures are bound to DNA, some to inhibitors of P53, and it's a complex thing. And there's many properties of it. And so one thing we've been doing is using what's called parallel coordinates, and you've seen a talk about this, to take features of the ensemble, map them in this case onto sort of more or less 20 dimensions, and then make that interactive and zoomable. So I can select, for example, all residues. This axis here, for example, has all the different macromolecules in the system. So there's DNA and there's other proteins. You can just select, show me all the residues that are in contact with, let's say DNA. Just I can make a selection there, and then show me all of those residues that are solvent accessible. So one of these, can't quite read the axis, but one of these is, or let's say in disorder, there's a disorder axis. So show me all the residues in contact with DNA that are predicted to have high disorder. So you could ask interactive complex queries like that, literally navigating through multidimensional space. So we have a prototype for this. It's not a query, obviously. It's something you have to install and run, and it's something we want to do more of. But that's really a prototype. I'm going to mention some crowdsourcing stuff we've been doing, which I think is really interesting. I don't know if it would be of direct relevance, but I think it's an interesting new thing that we've been doing over the last year that I don't think you've heard of before. One is, it costs so much money to determine a structure, and these are important. We should begin to take each individual entry also seriously, not just the collection. And one important question is the orientation that we present a structure. And you know, most people never click off defaults, and usually we generate a thumbnail for each structure, and that's how most people will ever interact with the structure. All they'll see is a thumbnail. So we better put some work into choosing a good view. If I've got a three-dimensional object and I show it to you like that, you can't see many details. So typically what we do in structural biology is we use principal components. So we sort of calculate the long axis, the second long axis, and we make the thinnest axis through the Z plane, so you minimize occlusion, which is a reasonably good strategy, but is it the best strategy? So we decided to test this. So we took a series of structures, 100 points of view around each structure, and then we take each of those pairs of those points of view and then present those pairs. This is the same structure seen from 2 points of view. Which of these two views do you think is giving you a better indication of the overall shape? And we ran that on a thing called Amazon's Mechanical Turk. Some of you might have heard of it. It's a service where you could actually ask simple questions like this to hundreds, maybe even thousands of people, and get a large number of responses, and then do real statistics on the response. This kind of thing has been done before with real-world objects like statues, and there's a bunch of different mathematical methods that have been devised to sort of predict what is going to be the best orientation, and people have tested those mathematical methods against crowdsourcing, things like that. One of the things that's emerged from that kind of study is a thing called viewpoint entropy, and essentially the way we've used viewpoint entropy is we took secondary structure, we took each element of secondary structure, regarded them more or less as equal, and tried to find an orientation that presents more or less an equal amount of information about each secondary structure element in the view. So this is very much dependent on representation. If, for example, you were to choose a surface, you'd get a very different result. What we found, this was a preliminary study which has just been accepted, we published later this year, across 4 structures, and comparing how, this is reconstructing the pairwise voting from a couple of hundred people in each case, and comparing that to viewpoint entropy scores. We found actually quite a strong correlation, better correlated actually than principal component analysis. So we found that principal component analysis for ribbon representation seems to be a better predictor of a good point of view for the structure. We have a larger paper in preparation where we've taken actually 43 structures. Actually, it's the first or the second level of Cath. So this is sort of all the different kinds of domains, and systematically predicted the best way to view each of these domains. Well, not predicted, actually. Ask the crowd what they think the best way to view those domains it would be. And so I think this is interesting and shows the power of using something like Mechanical Turk to really reality test interfaces, different ways of viewing things, really get feedback from lots of people. I think it's a nice idea. This is a very nice paper. This is Julian's work, not mine, although the concept was mine. So now a related test we've been doing is about the visualization of uncertainty. So we certainly have this problem in Aquaria, which is that in many cases, when you go there and you open up a structure, you're not looking at the experimentally determined structure for the sequence you're interested in. You're looking at essentially a model, and that model has varying qualities. It could be really good. It could be an experimental structure, or it could be something that's even down to like 8% sequence identity. In other words, 92% difference in sequence. And we wanted to give the user a very different experience. If you're looking at an experimentally determined structure versus looking at a really lousy model, we want to pre-attentively, before they've even thought about it, immediately communicate that there's something going wrong in the case of low quality models, and there's something going right in the case of experimental structures. So the concept was, this is a generalization of the problem of how do we communicate uncertainty. And the problem that typically happens when we visualize data is we run out of what we call visual channels. So you use X and Y, and if you're making a simple plot, okay, you can draw an error bar, right? So you communicate uncertainty with an error bar. But when you deal with spatial data like this, we're using all the spatial information. You don't want to start crowding the space with error bars because it distorts your perception of the structure. Okay, we can use coloring, but often we run out of visual channels. And so the idea was perhaps we could use aesthetics to communicate quality. This is just a slightly crazy idea, but the idea was that if you sit down and you look at an experimental structure, it's nicely colored and it's kind of shiny, and everything is good with the world. And then as your structure gets worse and worse, just this immediately pops up on your screen and it looks kind of mottled and not kind of clear, and there's this pre-attentive communication of there's something wrong. And we want to give people that sort of hesitation. So this was an idea of using aesthetics to encode data quality. And we tested this. And so this is part of the Aquaria coloring scheme. That's just what's built in there. Our driving force was that concept I just mentioned. And now we reality tested, did the concept actually work? So we took the same structure, same orientation, but colored it with these, this is the 100% coloring scheme, and this is what, 50%, let's say, coloring scheme. And then asked people, which of these do you prefer? And so people's preference on the y-axis and the sequence alignment on the x-axis. Overall, for these structures tested, we were getting a positive correlation, so we arguably had succeeded in encoding data quality in aesthetics. I think it's an interesting idea to be explored in other ways. Actually, when we showed this to artists, they wanted to print out the structures and have the residues that have the substitutions grow mold on them, or try all kinds of different texturing. So it's opened up some sort of creative approaches that we could think about using in the future. I don't know if I mentioned this last time, and I can't turn this down because it's HDMI. That was my fault. I asked for it. Is there a volume control anywhere?
Maybe on the remote.
On the remote?
This one.
Okay. So one of the basic problems that people have is just learning how to manipulate a structure. That was not a good idea. Well, it's okay. We don't have to. I can talk over it. So there's a thing called the Leap Motion. Costs about 50 euros. If you've got one, you can just plug it in and it'll work with Aquaria. So it's an alternative way of moving things around. I'm not saying it's the absolute be-all and end-all, but it can be impressive for setting up demonstrations and so forth if you want to impress your investors or your provostees. A Leap Motion. So it's a little.
You just need this little.
Yep.
I was gonna buy this with the money of your.
Yeah. So just plug it in and open up an Aquaria web page. About 50 euros. Yeah. There's some laptops with it built in now, so you can. Anyway, I think there's a lot of scope to do more, and we're beginning to look at using 3D printing. This is some beautiful stuff from Arthur Olson's lab where they. Do
you use it?
I have various printouts. I haven't. So this is what he calls the augmented reality toolkit. I haven't used it directly myself. I think it's a beautiful idea, so I just like telling people about it. Which is to. Oh, the Leap Motion. Well, for demonstrations, yes. Yeah. I actually have it in my bag in your place, so I can show it to you.
Yeah, but you're not using it every day.
No, not every day. I think it's for demonstration. I mean.
So
that's a whole other discussion.
Visualization for nice pictures, but I do it anyway.
Yep. Yes, we're almost at the dark protein. So this is a beautiful idea of printing out a protein structure and then using that printout as a direct proxy for an on-screen representation of the same thing. And then, because this is the thing that you're driving, you only need just a simple camera that can fit that, can recognize that protein at any orientation and directly drive this, and then do, for example, electrostatic fields. I think there's a lot we can do with with 3D printouts. And it's one of our goals is to connect a 3D printing service to Aquaria so that Aquaria basically helps you find structures and do various things with them. For example, coloring features. A beautiful next step would be to make it easier for people to actually put structures in their hand. So that's Aquaria. And here's, remember I said before that one of the themes from this talk it's going to come up is what's missing. So one of the things that's missing, we've been looking at this for years, is there's these regions in the sequence where there are no structures. And what we realize is it's a much stronger statement than just we don't know the structure there. With Aquaria, what we've done is systematically model all parts of the sequence onto all experimental structures. And so if there's anything missing, and with AJ Splits we believe we're using essentially the best method to do this, this is a stronger statement. This is saying that we know that that sequence is not detectably similar to anything that we've ever solved the structure of. So not only don't we know it, I mean, we don't know the structure of a lot of these regions, they're modeled, but this is something we can't model. So something weird is going on there. So we call it a dark region, a region where structure is unknown. And our idea was to sort of look at these regions, take them seriously and try to characterize them. This was kind of fun, trying to map the percentage of the human proteome that's known onto Julian's baby. So. And so to step you through it, so for each sequence in Swisspot, so there's 500,000 well-described protein sequences, we found the regions of the sequence that are in the PDB. Then there's regions that are not in the PDB but are in PSSH2, so we can say something about the structure, although we don't actually know the structure. That's important to point out. So we have some information about structure. And then there are the regions that are left where we have no information about structure. We call them dark regions. And in some cases, the dark region extends across the entire protein, and we call it a dark protein. If we take all eukaryotic proteins in Swisspot, 170,000 proteins, line them up end to end, and ask the question, what fraction of amino acids are covered by or are in the PDB? It's about 4%. That's the 170,000 eukaryotic proteins in Swisspot. And based on that 4%, we can, using PSSH2, extend that to around about 50% of the proteome. We can tell you some information. It's quite subtle. There's a subtle data visualization here that this is sort of knowledge, although really, if you have a PDB structure, does that mean you know everything about it? Not at all, right? It's just we have one piece of experimental data. So we don't really know the 4% one completely either. And then some of the things like that are 99% identical, and we know those, but then there's a fall off, right? And you get sort of less and less certain. At some point you have to draw the line, and 2 master's theses were spent here working out where to draw that line. And then that leaves about 50% of the eukaryotic proteome where we just have no structural information, and about 1/3 of that is contained in proteins that are completely dark. No part of that protein matches onto anything in the PDB. Dark proteins.
This is not from you. This is Swisspot, right?
This is Swisspot, 170,000 eukaryotic proteins.
That's very good.
Sorry?
The view will be very different if you map it to a proteome.
Yes. Well,
Yes.
Yine. I mean, if we compare it to Michael Levitt's 2009 paper, I think overall it wasn't that different. But of course, there's definitely biases in the database. It would definitely be interesting to do that. We haven't done that yet. So, if we look at other domains of life, so it turns out that we're pretty similar to viruses, about 50% unknown. They have a larger fraction of dark proteins. And bacteria and archaea, interestingly, we know less about them structurally, like between 2 to 4%, but we can extend what we do know to a much larger percent. These are simpler forms of life, and they tend to reuse the same architectures. There still leaves about 12, 13%, this sort of stubborn fraction of this proteomes that we just don't understand at all.
By the way, this number is also, if you look at proteomes, this cannot be.
Yep, as I say, we haven't looked at proteomes, and that's definitely what this paper has opened up many next steps, and we have not taken most of them, and we're going to take some of them. We're going to take some of them together, you guys. Other people are going to take. There's just lots of things to look at. I'm going to just drill a little bit into details. So some of you might have seen the paper, but this is something in the paper it's not made a very strong feature of, and I think it is a useful idea. We had a lot of useful ideas, I think, in the paper. So one of them is what we call a darkness profile. So these are, again, eukaryotes, and we're looking at all the 170,000 eukaryotic proteins, and we're asking how many of those proteins have a darkness, let's say, between 0 to 25%, have like a low level of darkness. And actually, a lot of them do. So the area under the curve is essentially the fraction of total proteins there. There's this fraction at the minus percentage because of the smoothing we're doing, right? So most proteins, most eukaryotic proteins have sort of a lowish level of darkness with a median, I think it's around about here, and it falls off. But then there's this peak of proteins that have 100% darkness. We turn that around and then take that median 28% for eukaryotes, and then put it on its side. so now we have the darkness value. Now let's look at individual proteins. So these are 170,000. And for each protein, measure also a disorder value. And this is disorder measured by IU-PRED. We find something very interesting, which is that most of the proteins in eukaryotes fall above the diagonal. And that means that darkness is greater than disorder. And that means that most of the dark regions are not disordered. That's kind of, and these are, what we're looking at here are proteins that are, where the whole protein's not dark. So we're only looking at this region here, less than 100% darkness. 100% darkness, the real dark proteins, they're all crowded up in that line. So in this graph, you can't really work out what's going on with dark proteins, only the darkness or intermediate levels of darkness. So what we did is another plot here that it compares the dark proteins compared with everything else. And so these are dark proteins, and these are everything else, including proteins that are like 90% dark. They're all in that non-dark region. So overall, the surprise to me was that the levels of darkness between completely dark proteins and proteins that are not completely dark is relatively similar. 6 to 10%. For eukaryotes, actually for prokaryotes, it's the other way around. That non-dark proteins have a higher level of disorder than dark proteins, just slightly, but it's weird. I don't know if we'll get to that, but there's a lot in here, and again, I feel with this paper we're really just scratching the surface. It opens up so many questions. I mean, this entire analysis could and should be repeated with different kinds of disorder predictions, for example, and there's, again, so many more things to do. But the key surprise, and this sort of summarizes a result that was implicit in the previous graph, was that if we take the dark proteins, now these are only dark proteins, and this is not a perfect graph, but it shows something very interesting. Look at the proteins that have more than 25% disorder, and it turns out about a third of dark proteins have high levels of disorder. All the proteins that have a high level of transmembrane, and this is sort of like a Venn diagram, it also shows overlap. So we have here proteins that have a lot of compositional bias, and they tend to overlap with disorder regions. That still leaves about half of dark proteins with low levels of disorder, low levels of transmembrane. In other words, we don't really know why they're dark. We don't know their structures, and we don't know why we don't know their structures.
Other than short proteins?
Next graph. And that unexplained fraction, or the unknown actually increases for the other domains of life. So most dark proteins, we don't know why we don't know their structure. And so we then started to test a number of other hypotheses, like length, for example. And it turns out that indeed dark proteins are shorter. That's consistent. This is just eukaryotes I'm showing here. But not like massively shorter, not most of them. But they're significantly shorter, that's for sure.
Do you have additional information on the sequence level for this information, or is there also no annotation, or is sequence similarity to known proteins?
We'll get to that too.
Yep, next slide. Or the one after the next slide. Oh no, this slide. This is the slide. What am I talking about? So this is exactly that question. So these are dark and non-dark proteins, and this is the number of matches against UniProt, so the full 60 million. And so a non-dark protein typically has 400 or so matches compared with 27. For eukaryotes, I think these numbers, the difference gets even greater for I think viruses and various others. So it's big difference in the number of matches, matching sequence.
Have to be able to subtract the trivial. Non-trivial.
What do you call trivial?
We can discuss that.
Okay. So then this...
to make it clear for that one, so this is not individual proteins we're matching, but it's like the families in the hated split database. So it's clustered a bit by if the sequences are really similar, and that takes out things like, I don't know, E. coli sequences, which are really, really similar to each other.
Yeah. It's already it's got a lot of non-redundancy has been removed already. So
yeah.
So you understand what my statement, the background of my statement.
No.
So ultimately, the ones that remain to be discovered are the ones where we cannot recognize similarities.
Yes.
So for larger families, the probability that I cannot recognize similarities is much smaller than for smaller families. And that's the trivial background.
Right.
And somehow we have to normalize that out.
Yeah.
And I don't know how.
Yeah. there's still these guys. There are dark proteins that we have a lot of families, and there's some interesting things there. That proteins that we know a lot about, but for other reasons we can't determine the structure. This was the second big surprise, and I have to. Okay, so these are I'll just give the data as they were. So basically, just went into string for each protein and looked at how many other proteins is this protein known to interact with in string. And again, this is all eukaryotes, 170,000 proteins. And we see sort of an overall peak in 110. That's the ribosome complex present across many organisms. There's, ribosomal proteins are there, and they have about 110 interaction partners. Dark and non-dark, big difference for non-dark proteins across all 170,000 eukaryotes in Swissprot. A median of 10 other proteins where we have high quality information that they interact with 10 other proteins at least, and then the median for dark proteins was 0.
Is white similar to the entire string?
The entire
string.
Sorry?
If you just forgot about your labels dark, not dark,
yeah.
You took the entire string, would that look like white?
Yes. Yeah, white is larger, so it's only about a fifth of proteins are actually dark, because a dark protein it's a strong statement, the entire sequence length. So the majority is actually this. The way these things are scaled with these density plots, you don't notice it because we're trying to we balance things out.
Wait.
So
maybe you answered my question, maybe not.
I think I did.
But so there are proteins in string that have neither level dark nor non-dark. They are not in Swissprot, for instance. There's one simple way in which that would happen.
Yeah, we're not showing them here. So exactly. We're only showing the 170,000 where we have darkness and non-dark scores. But string is mostly Swissprot. I mean, it's well, it's even less than Swissprot actually. It's pretty restrictive.
Okay. So I really wonder how that would look without any structural constraints.
I'm pretty sure it looked very similar to the non-dark. Yeah.
Really?
Yeah. And then the other surprise was that when we looked at the regions.
Cannot be.
Why?
Because PDB stands out in every biological feature we look at. And non-dark is essentially PDB.
No, there's a big difference between PDB. PDB is 4%. Non-dark is. Yeah, I'd say to my mind, they're very, very different because PSSH2 goes from PDB to a much larger set. Strict PDB is really a small subset of the whole thing.
Good.
The other surprise was that when we looked at which regions of the cell have this, where dark proteins are annotated. Well, first I have to say that truly dark proteins, and it's what we're looking at here, tend to have no annotation of location, no annotation of other functions. That there's a strong tendency, in other words, what we call structural dark, or lack of structural knowledge correlates with lack of knowledge of many other properties. But some of them are annotated in Uniprot, in Swissprot, as having some location information. And when we look at those and we look for overrepresentation, we see that the locations in the cell that are most strongly overrepresented with dark is actually the extracellular space or amongst secreted proteins. Well, I wouldn't say it's because they're short. It is consistent with them being short. Yeah.
So no.
It's consistent.
No. Short proteins are easier to do the structure of the long proteins. So the because certainly is not right.
Well, it's consistent.
Short and disorder.
Yeah, I mean, secreted proteins also tend to be short, so it's a.
Yeah, but again, so they should be non-dark because of that. So the expectation would be the other way around. Yes. So, and
one thing I want to make clear is that this is not, most dark proteins are not secreted, right? In fact, all right, I'm going to go on to the next graph, which is about function. So we looked at functional categories, and there's a little hard graph to understand, but I'll interpret it. We looked at functional categories that are over or underrepresented amongst dark proteins, right? And the number one result is what's not shown here, which is that the vast majority of functional categories are not overrepresented in darkness and not underrepresented. That means the dark proteins are everywhere and they do basically everything. It's a selection of proteins, right? But there is a subset of dark proteins that seem to, some of them are secreted, for example. That is the strongest signal we've seen in terms of their location. So it's a subset of them. And this is a generalization of that, which is now not looking at location in the cell, but looking at other properties. And again, these are just functional terms derived from proteins from Swissprot. and then we look for those terms over or underrepresented. What we've done in this plot is, so these are, for example, all the terms related to tissue specificity. If there's any term that's overrepresented amongst dark proteins, we size the cell here according to the p-value. So we're using a p-value to estimate how much overrepresented it is. And this is sized according to minus log p, so a large area indicates a strong overrepresentation for dark. A bluey area is a strong underrepresentation for dark. So dark proteins are overrepresented in all these tissues. We can see some very prominent tissues. There's other tissues we can't fit the labels on there, but we have the file if you want to download it. Lots of other tissues. Essentially a bunch of specific tissues, particularly tissues related to secretion. Secretory tissues, milk glands, skin glands, saliva glands, venom glands. And the only 3 tissues that were underrepresented in dark were red blood cells, which are almost entirely cytoplasm, where dark proteins are underrepresented. And 2 other tissues. One tissue was called, the tissue is ubiquitous, and the other tissue was called widely distributed. The tissue has been widely distributed. So dark proteins tend to be, or there's a subset of them are expressed in specific tissues. Part of the reason probably why those guys are dark, because they only occur in specific tissues. You only see them perhaps in specific times. We found some evidence that some of them are only occurring in certain times of development. And then we got an association, we look at domains. Many of these domains contain multiple disulfide bonds. We see a strong tendency that dark proteins have more post-translational modifications or specific post-translational modifications, cleavage and disulfide bonding most prominently. And then, for example, the annotation of specific catalytic activity is underrepresented in darkness, which makes sense because you can generally only assign catalytic activity to a protein if you've got some information about its structure. So all this sort of made a consistent story, which is that story that we put together in the PNS paper, but as I hope is coming across, that it's really the sort of the tip of the iceberg. Essentially any question that's been asked in bioinformatics about proteins, you could re-ask the question, how does it compare dark versus non-dark? So there's a lot to do, and I'm not going to do it all, so there's plenty of stuff out there. One of the papers we're working on next is the dark human proteome. Actually, a lot of the stuff we had ready, we couldn't fit it into the PNS paper. So this is the distribution of darkness in human. This is the human proteome. We know about 12% of the proteome. This is looking at the amino acid fraction. About half of it we can predict something with PSSH2, and then half of it is dark. And dark proteins cumulatively account for about 16% of the proteome. It's a larger fraction of the proteins, about 20% of the proteins, because dark proteins tend to be short. So it's 16% of the proteome, but about 20% of the proteins. And that's what the function plot looks like for humans. It's completely dark, and there's interestingly some association we see with certain kind of cancer and certain kind of retroviruses. Sorry, these are all the functional terms that are over or underrepresented in darkness in amongst human proteins.
And over means black, and under means I don't see. Or
There's nothing there. There's nothing underrepresented.
Okay, so they're all over.
They're all overrepresented for dark, yeah.
Okay.
Yeah, but I mean, I would say everything we've done up until now, I was saying to Burkhard, and I think also to Andrea, we've just gone for the low-lying fruit, right? All I've done, in most cases, is we just looked at SwissProt annotation and nothing else, and not even all of the SwissProt annotations. We're flooded with data, and p-values are ridiculously low, like 10 to the minus 250 you saw before. Sorry?
CDS?
Where's that?
CDS.
Where?
Dubious CDS prediction seems to be the most right.
Yeah. So caution. So this was one thing we tested in the paper. Actually, it's in the PNS paper. And clearly, one possibility is that dark proteins are not proteins. They're long non-coding RNAs or something, pseudogenes. And indeed, we see an overrepresentation of cautionary annotations. But then we did some checks, because I actually worked next door to a group who studied long non-coding RNAs, and we looked and did some predictions trying to say, is there evidence that they actually are long non-coding RNAs? And the evidence is maybe about a couple of percent of what we call dark proteins might be long non-coding RNAs, but it's, in other words, that seems to only account for a very small fraction. I mean, the point is we've built this on SwissProt. Every one of these 500,000 proteins are manually annotated, and it's not the gospel truth, but it's the best database we have. So, but these probably are proteins, but they don't seem to. We don't know the structure, and we don't know other things about them.
So looking back at the secretion story, is that similar to what you had before? The prevalence of the secretion signal that we see here?
Secretion signal, well, in terms of tissue...
It's less, right?
testes only. Testes only. This is what I'm saying. So previously we're looking at 170,000 eukaryotes, and so the numbers are larger, statistics are better. And what I was just saying about this is, I would say, it's a very crude measurement. Right? This is just what we have done. I mean, Tanya has given us for human alone has given us the prediction of location, and we've gone through that for those and found that secretion is I think the strongest signal again. Secretion, endoplasmic reticulum, and Golgi apparatus, which are all on the secretory pathway. Unfortunately, your data can't distinguish between them. So again, this is crude analysis. Your prediction method only gives us the one location as the best shot. It doesn't give us a ranking of locations.
It doesn't.
Why not? You could.
Because don't predict multiple locations.
Yeah, but you could.
Well, it was explained to me that fundamentally you don't have those data. You only have single location data as input. So if you could give us a ranking of the tissue, that would be great. Yep. So.
These annotations, are they from SwissProt or?
These are SwissProt annotations only.
Have you ever normalized them somehow, or you just put them into the analysis?
When you say normalized, what do you mean by that?
For example, localization. If you need chirals between locus and cytoplasm, it's not that every protein that is localized in these two compartments would be annotated like this. Because the annotations, they really sometimes random.
So all this is all human proteins.
No way, just use your prediction method.
Yeah.
So we have to figure out our second best. This is a very good.
This is a good idea. Break it down, and but again, I think the best way is SwissProt is, to my mind, sort of high accuracy, but you're not getting that much precision. I'm sure you could predict a lot more if you use more.
That it's human annotation in the very end, and they're not consistent.
Yeah, no, I totally agree with that. So here's some really new data. So this is from the Proteome DB in the Munich team, looking at all the proteins that are expressed in what is it, 100 and something tissues in the body, or 70 tissues. So for each tissue, they've got a list of proteins and a level of abundance. What we decided to do, just kind of for fun really, was to take each tissue, look at all the proteins in that tissue, and then let's say it's the brain. Take the brain and look at the proteins that are highly expressed in the brain, and then the fraction of each of those proteins that are dark, and then you could calculate a darkness value not for each protein but for each tissue. So which?
It's not the surprise value.
The surprise value?
So this is per residue. 35 is the number that you expect.
This is the per residue value. Oh, you're seeing the fraction. Good. This is the total value, which is 50% is what humans have, about 50% darkness, right?
50? Wasn't it 35?
No. Total darkness is for humans actually it's a little bit more than 50, about 55.
So the first one would be expected, and the others are underrepresented.
Well, so one of the things is, and this is the subtlety of the experiment. These are very different data. This is derived from proteomics, and in the proteomics experiment, you don't measure everything that's in SwissProt. So, for example, many of the dark proteins are not listed as occurring in any tissue. So dark proteins overall are underrepresented amongst the mass spec data. One could call them the dark proteins of mass spec.
They have all 20,000. I'm totally confused. You have the same data set?
Not at all. Not at all.
But you could use the previous one, the 50% with the same data set. They have 20,000 proteins.
No, they don't have all. They don't see all.
No, not at all.
There are some that they don't see.
A large number.
How many do they not see?
I think about
A quarter, I think. Yeah. Yeah, and most.
But the Nature paper is about we saw 20,000 proteins.
No, read it. They don't.
Oh.
I mean.
So they only see 15,000?
I think it's about. Yeah, it's about that. It's about a quarter.
So now let's get back to the 16.
And of the ones missing, many of them, the majority are dark proteins. So it's about abundance, right? Because you've got the thresholds.
The more so you should normalize this somehow.
Well, yeah, we've tried different kinds of normalization, but the thing is, okay, the tissue in the body that has the highest level of darkness is the heart. Which, and I don't want to make a big point about it, it's not any, actually one decimal point is not different from the others. But that's interesting because, of course, heart is the tissue that is associated with heart disease, which is the biggest killer. Actually, I don't have the data, but we recently did another experiment like this using totally different data, looking at actually, what's that database, the OMIM database. So looking at UniProt entries, and some entries are listed as having a genetic link to a disease, right? So it's a genetic base of some diseases. and then categorizing the diseases, and so looking at the disease that has associated with proteins that have the highest darkness, so the darkness value of each disease. And the disease actually with the largest level of darkness was cardiovascular disease as well. So that was totally different kind of data that was interesting. So that this dark human paper we're working on, I think this is the, I wouldn't say it's the killer blow. It's not going to, obviously not going to make nature or anything, because it's not like a hugely strong link, but it's definitely pointing the direction towards medical significance or clinical significance of the dark proteome, which was kind of obvious that it does have that, but this actually gives it a more of a solid basis. So, and gives the paper kind of a focus. That's the ones at least. And then something, I don't know if I haven't presented this before, was looking at dark gene clusters. So you can take each protein and you can map it down onto the gene where the protein comes from and map them down onto chromosomes. And if you do that, if you think about it, you're going to get, let's say for one chromosome, you get a string of genes next to each other, and each gene either codes for a dark protein or it doesn't, right? So we have dark and non-dark genes. And in some cases, what we thought was, if groups of genes all together code for dark proteins and they occur in clusters, this could indicate kind of common function. And so there's a bit of stats behind this, but we actually found, well, there are a lot of those clusters. These are the ones with really low p-values. And it's interesting, this table, this is for obviously human across the human chromosomes. It actually takes you through a bit of a smorgasbord of what dark proteins actually are and what they look like. So we've got, for example, a whole bunch of keratin-associated proteins, two dark clusters of those. So these are, keratin, skin, hair, and so forth. And we know about these proteins, we know about their functional roles, and we don't know their structure. So these are things with lots of relatives, but with an unknown structure, but actually relatively known function. And then we have things like this, this is a cluster where we get a whole bunch of proteins associated with saliva, secretion, digestion, and so forth, mouth. And these proteins you might not have thought were connected, but they sort of fit in with that pattern that we see amongst the secreted dark proteome, particularly into the gut and into the secretion and so forth. So it's sort of connected, or darkness explains it. You've got a cluster here with transmembrane proteins, because we know that some dark proteins are indeed transmembrane, and here's a nice little transmembrane cluster. And then we've got these two clusters in the X chromosome. Many of these proteins are actually unique to human currently. There's no other organism known to have anything similar to them. So these are really kind of the totally new, weird proteins that we don't know anything about. So it's an interesting sort of smorgasbord of dark proteins, and these clusters seem to be significant. Well, they definitely are significant according to the stats we've done. Interesting to look at.